Chris back to talk to us. Chris is a proud South African trained at UCT. I sent round in the email a short bio for Chris, so I'm not going to go through everything, but he's an amazing and amazingly accomplished and a prolific trialist who currently heads the primary care, or professor of primary care in Nuffield Department of Primary Care in Oxford, and directs the clinical director of Oxford University's Primary Care Clinical Trials unit. And last year, he spoke to us about new platform trials for COVID vaccination. And Chris agreed to give us an updated talk now, focusing on PANORAMIC and the issue of antimicrobials for respiratory infections in the community, for which he's globally recognised as a major leader. So Chris, it's absolutely fantastic to have you to talk again, and welcome, and the floor is yours.
Okay, thank you very much. Just trying to sort something out. So yes, thank you so much, Mark, for those very kind words. And you know, as I mentioned the last time, I was kindly invited to be with you all. You know, I'm a sort of an honorary member of Mark's department, so really proud of that. Given that it's my alma mater, it's a really special affiliation that I have, and hope to continue it and continue to work and interact with all of you. So I'm talking here about a panoramic view of transforming the design and delivery of large-scale randomized controlled trials of antimicrobials for respiratory infections in the community. And so I'll sort of talk a little bit about bacteria as well as viruses, and the ways in which we get evidence, which I think we need before we use these drugs at scale. So I'll start off by taking you back to St. Mary's Hospital next to Paddington Station in Preed Street, and you can see the blue plaque there. I think Mark will probably know the story better, but one of these windows at the top there was Fleming's office, and I think it's one of them in which the mold kind of wafted in and landed on his Petri dish, and that's where he discovered penicillin. And actually, in the building just to the right there's a foyer, and I took this picture of Fleming with my iPhone, so it isn't just a download from the web. And you know, he made the point that, well, he said the thoughtlessness of people using penicillin unnecessarily got to take responsibility for people who die from resistant infections. And so that's quite a serious thing to say, but it might be true. I kind of mentioned Fleming as perhaps the second most important graduate of St. Mary's Medical School. And I'll leave you to make a judgment, Mark, about who the most important one is. Certainly the most important one in this meeting is not Fleming. Now, another blue plaque is outside the building, actually, where I work at the University of Oxford. And here I am with two of my classmates graduating in 1986, below the penicillin plaque there, which says that's in that building that the first patient received penicillin. And these are the actual notes that were made at the time of this Mr. Anderson, who was the first human to receive penicillin. And it's kind of, I know, it just tells a story that's worth reminding ourselves of. And the notes reflect here on the 11th of February, this very sick man septic side of his face, and they basically drained his orbit. A large amount of pus was released. That was on the 11th of February. The 12th of February, they started penicillin. As early as the 13th, they're saying much less discharge from the scalp and orbit, and right eye has clear vision, and conjunctivitis has subsided. By the 15th, the patient is very much better. And by the 18th, they stopped treatment of penicillin. So they didn't have enough. And as I understand it, they were trying to get more by purifying it from his urine and giving it back to him. But that unfortunately ran out as well. But he stayed well. On the 20th, he was afebrile. By the 5th, though, his scalp began to discharge, and he was much weaker and frailer, and became jaundice on the 9th of March, and died a few days later. So I don't know, I just kind of thought, reminding ourselves, not that anyone on this call needs reminding, but you know, the power of these antibiotics go back to that first case, and we misuse them at our peril. And we landed in a whole lot of trouble here with the, call it the parallel pandemic of antimicrobial resistance. And that's because we practiced assumption-based medicine, and we made an assumption that, you know, if it's good for somebody like Mr. Anderson, then penicillin is going to be good for people who are less sick, and even then for prevention. So if it's good for pneumonia, probably good for cough, and therefore it's good for snotty kids and earache and so forth. And we didn't do the trials in the places where these drugs are used most frequently. For example, you know, it's only recently that we've been doing proper randomized control trials of coughing patients in the community, and we show numbers needed to harm very similar to numbers needed to benefit, and that benefit is perhaps only by a day. But you know, being a GP, and I guess many people have different views of GPs, but over here we often considered a bunch of whingers. We'll notice that it's in the community that we treat the vast majority of patients. 95% of healthcare contacts in the UK are in primary care, and most of them are the patient's problems are sorted out there. But the trouble is the funding, the kudos, it's all gone to the clever guys in the hospitals and in the ICU patients. So if you're snatching somebody back from the jaws of death, I guess like Mr. Anderson, that's going to create a lot more interest than, say, preventing somebody from getting into that ICU in the first place. But it's probably in the community where people have the greatest reach and impact of these antivirals and antibiotics if we use them properly. Let's go back a few years to the swine flu pandemic. Here we had the opportunity now to start using antivirals in an evidence-based way, really, for pandemic illness. And what we did in the UK, at least, we gave this drug out in industrial volumes to people. We said, you don't even, you know, you shouldn't come to the general practice. Just if you've got the syndrome, send a friend, a flu buddy, to the pharmacy and pick up a supply of Tamiflu and take it. Now, we didn't randomize a single patient then, so we still don't know really if that drug helped these people with swine flu. We certainly know by giving out the drug without examining people that we did a lot of harm, but you know, we don't, and this drug remains one of the most contested drugs on the planet, really, and is largely not used, certainly in the community, because I think the evidence is unclear. And if we had randomized people, we could have gotten an answer fairly quickly. But this just makes the point about keeping drugs to treat people who've got a good chance of benefit and keeping them away from those who've got a poor chance of benefit. So really the whole kind of research effort that I'm talking about is really that 2x2 table about targeting or personalizing medicines, not by etiology so much as by chances of gaining benefit from the antibiotic or antiviral. And this is how it was really prior to that swine flu. In fact, after the swine flu epidemic in the 2000s. And, you know, you get a pandemic or epidemic emerging, you get, you know, rapidly the public health people get involved, and then you get, you know, observational research, preclinical research, and it's towards the end that you finally traditionally start to get some randomized trials. And if we had done those randomized trials early on, we wouldn't be uncertain about whether Tamiflu worked meaningfully against the swine flu. So we're trying to get a different model here, where you get the public health response starting quickly, but then we get the clinical research response happening early on, so that we can generate evidence from within the pandemic to treat people in that same pandemic, so that your intended use population is the population in which you have gained your evidence. So with this in mind, we thought, well, we'll do a peacetime study, if you like, to test the systems, develop the systems so that we could rapidly set up trials should there be pandemic. So we did this trial of oseltamivir for influenza-like illness under the PREPARE network of excellence that was funded by the European Union. And I managed over 3 winters to, in I think 10 countries, randomize 3,259 patients. And you know, the interesting thing was this was an open trial, so it wasn't placebo controlled. It was usual care versus oseltamivir. And we got exactly the same overall estimate as what was obtained in placebo-controlled trials. And you know, a lot of controversy about the systematic reviews around oseltamivir, but they all got about a day's benefit, whichever way they skinned that cat. And what we found, yes, exactly the same thing for the overall estimate of that entire population. But because of our trial design, we were able to power this trial in a way to look at benefits by subgroup. And we found really that the older people, as you would predict, who were sicker and who had more comorbidity tended to get benefit, and in fact children as well, more than a day, and they started to get better like 3 days sooner. So that brought us a bit closer to the vision of randomizing patients. You know, we could show that we could get these trials up and running, and we learned a lot about doing that quickly. So when the pandemic emerged, we sort of thought about a platform trial based on that experience, and you know, realized that, you know, it's as important as it was to find treatments that help the sickest people in hospital, perhaps in the community, if we got stuff that worked for early treatment, that might be very useful. But it was also very useful to show what didn't work. And as colleagues will know, people were giving out antibiotics a lot for COVID around the world. And particularly in the UK, they were using a lot of azithromycin and doxycycline, because in the lab, apparently they found that these antibiotics were killed the virus. There was a thought that many of the people who got pneumonia from COVID might have benefited from these antibiotics. And then the anti-inflammatory component was potentially helpful. So I suppose partly out of desperation, a lot of people were getting these drugs. And going back to my 2x2 table, we didn't have the evidence there as to who was going to benefit and who didn't. At that time, there were numerous small trials, single drugs that weren't recruiting to target, and could never. And there was a lot of nationalism. Every country had to have their own little trial, or their own trial, or several trials, and every university, and that sort of problem of scratching in each other's salads to try and get the patients into a whole lot of trials that couldn't really deliver. And in fact, I think there was at one point 120 trials of hydroxychloroquine on the registers, and wouldn't have worked out to. You couldn't power each of those drugs or recruit enough into each of those trials to get an answer. So we figured out that we needed to rapidly initiate a trial that had adaptive features so that we could add in treatments, because although we started with hydroxychloroquine, we didn't imagine that would be the answer only. So we knew there were other things coming down the line. We knew we wanted to add in antibiotics. So an adaptive trial would allow us to put in new treatments in series, as well as test them in parallel. And we didn't at this point seek to do what you might call a regulatory trial to determine whether the drug is better than a dummy version of it. We attempted to find out whether treating people with the drug did more good than treating them without the drug, if you see what I mean. So the question for a GP sitting in a consulting room somewhere is, am I doing this? Am I doing good by treating this patient with a drug as opposed to not treating them? So we felt that the comparator should be care without the intervention. And we can talk about that in a minute. And I got the nod really to go ahead with this trial on the 15th of March while I had COVID, and went on to produce a protocol for like 4 or 5 days later, and got ethical approval 10 days later, and had the first patient in a few days after that. So that's not a time I think about with any pleasure, but it shows that you can, under the right circumstances, with the right will from the approvals agencies, get going quite quickly. Doing research at scale in primary care, not so easy. We try to do this as a low burden trial, and we started out setting up GP sites, and there was we managed to set up sites about 800 very quickly, and getting drugs to them. They had to repackage it each time they saw a patient. And as soon as we, and all the contracts, you can imagine what a nightmare that is. And by the time we'd set this up, the government then said, don't go to your GP if you've got COVID or think you've got COVID, phone a helpline, which was called NHS 111. So we tried to recruit that way, but failed. But that got us thinking about central recruitment, which I'll say a little bit more on. But before that, I just want to go back to the issue of trial design, because that's one of the things that I was sort of thinking might be of interest. And, this is the issue about being able to do large-scale trials in primary care, I think, is thinking about different ways of doing it. And, when you do the sort of traditional two-arm fixed proportion allocation trial, which really wasn't appropriate for our circumstances, but there you do a sample size calculation, which to my way of thinking is a bit like throwing the bones a bit, but you hit on a number that you think will show a meaningful and clinically important difference. And you keep going till you hit that sample size, and then you analyze each arm by those who respond and those who don't, and then determine if there's a difference. But the problem with that is you might have got your answer earlier on, or you might have needed a few more patients to get your answer. And the sort of both the problem of type 1 and type 2 error become quite important then. So other ways of doing it, particularly if you want to not just have one trial per drug, is what I've mentioned already, is the sort of platform trial design, where you could have a number of drugs in the trial, both sequentially and in parallel, and then recruit people with different characteristics. And so you get a comparison both by drug and by people with different characteristics, because that 2-arm type trial that I showed you there can be limited sometimes, particularly if you're in the pragmatic space, because the GP will often say, all good and well, you guys doing that kind of trial in the hospital clinical research facility, but my patients aren't like that. You've excluded everybody with condition A or taking drug B, but most of my patients are on a lot of drugs, and what does that mean to my patients? So you can include a wide array of patient types as well as quite a lot of drugs in the platform trial design. And this contrasts with a basket study where you get a contrasting characteristics of patients, but a single treatment, or an umbrella trial where you can get a single type of patient with lots of drugs. But to go back to the platform trial, here you can see we have drugs A, B, and C and a control condition. In our case, it was usual care. And you can add in, as we go along, drugs D and E. Quite quickly at your first interim analysis, you might find that drug C has achieved a threshold for futility, so you can chuck that one out. You might suddenly think, particularly say with a viral illness, you better worry about resistance or mutation. So let's have 2 drugs. You combine them now, and you've got another arm of a combination. At the next interim, you find drug B is superior. And what do you do now? Because people know that you found a drug that's good, but it might not be that everybody could take it. So your trial doesn't need to stop now, but it could become part of usual care. So the drugs that are still in the trial have got to beat standard of care plus that drug that has been shown to be beneficial in that platform trial. Now, just about trial delivery, in the UK, I've mentioned that there's 8,000 practices, and you can't set them all up for research. So people are limited by where they live or which GP they are affiliated with in terms of their ability to contribute to research. And back in the South Wales Valleys, in that village there that you can see called Glencorrig, there's a famous GP researcher and practitioner called Julian Tudor Hart, who formulated the inverse care law. And he said that the availability of good medical care tends to vary inversely proportional with the need for it in the population served. And interestingly, he said this inverse care law operates more completely when medical care is most exposed to market forces. Nevertheless, we sort of thought about this and thought of the inverse research participation law, which seemed to be saying that access to research is often inversely proportional to a participant's potential contribution and to where the research findings could be most applicable. Because we're finding people join trials in big cities, but next to big health centers, but it's often in the most deprived areas of the UK where COVID was hitting hardest, in areas where practices were least likely to be involved in research. There was a lot of things that helped us with setting up that principal trial. We had nimble funding that we could apply for, and peer-reviewed funding that we used. The NHS set up a thing called the Urgent Public Health Platform, where they controlled the valve, shall we say, into the NHS. So not all 120 trials of hydroxychloroquine could recruit in the NHS. And they prioritized 3 national platform trials. So REMAP-CAP was in the ICUs, the recovery trial in hospital, and the principal trial primary care. And so there were numerous people with numerous exciting drugs they wished to trial, but they couldn't set up their trial basically unless it came through the urgent public health panel and then through a panel which examined the plausibility, shall we say, and the proof of principle for the drug. And we developed an online consent process and a way in which people could sign up irrespective of where they received their care through the web. We could actually text them if they had a positive COVID test about the trial, and they could be couriered medication if they were randomized to receive it, and we could follow them up online and by telephone. So in fact, sick people with COVID could participate in the trial with some of the drugs if without ever having to leave home. And this is the 7 drugs that we had in the principal trial. And I know colleagues there will be interested, particularly, I suppose, in what we found about ivermectin. But we've now closed the ivermectin and the favipiravir arms, and are following. We'll have followed those patients up for 12 months in July, and we plan to include the 12-month follow-up in the publications, because I suppose that's going to be more unique and more interesting. Does acute treatment with these or any other drugs impact upon the longer-term symptoms? It's these 2 papers that might be of most interest to colleagues on this call, where I think we were the first to show in primary care that I can see I've messed up a little bit. The left-hand side is supposed to be a paper about azithromycin, and the right is supposed to be about doxycycline, but in my haste, I've given you both. But you get the point that, particularly the doxycycline, there are others who subsequently did trials of azithro in primary care, but I think we were the first to show that. And colleagues from different jurisdictions have told me that they've, taken this trial to their colleagues and said to them, look, it doesn't work for COVID unless there's, of course, other indications. And in the NHS, it's quite interesting, we always worry about impact, but the chief medical officer can send out a clinical therapeutic alert, which goes to all doctors in the NHS. And this evidence was put out. So the next day, shall we say, the chief medical officer told, based on stuff from principle, but also recovery, that these drugs shouldn't be used for COVID in the absence of other indications. So in that kind of joined up health system, we were able to have an impact. So based on that experience, the government then, having procured many millions of courses of novel antiviral drugs, were wanting to deploy these drugs, with COVID really going like wildfire through the population. But to the credit, particularly, I think, of Jonathan Van Tam, who's a deputy chief medical officer, and Chris Whitty, the chief medical officer of the UK, they said, remember Mr. Anderson, and remember assumption-based medicine. And we deploy antivirals and antibiotics at scale in the community without doing trials at our peril. So they put out a call for funding, and based on that, we were able to get that funding. And a lot of people said to us, look, why do you want to do a trial? Roche and MSD have done trials, perfectly good trials, although they are controversial. Give it to the people who are sick. And it was somebody from the Telegraph who said, you're asleep at the wheel here. You need more of this stuff. Get it out there. And at the same time, there were other people who were saying, look, most people with COVID aren't going to get sick, that badly sick. Don't use these drugs because of the way they work. They cause mutations in the virus. There's all sorts of risk there. Don't use them. So a lot of controversy, but perfect equipoise, shall we say, in which to do a trial. And, you know, the politicians, it was the most politicized trial that I've ever been involved in. Politicians wanted the answer tomorrow. And, you know, Secretary of State saying there's 100,000 people with COVID today, and all you need is 10,000. I mean, why not give me the answer by next week, sort of thing. Now, we also were charged with doing a virology sub-study and a health economic study, as well as seeing whether this drug kept people out of hospital, or these drugs kept people out of hospital. And that was the main issue for the health service and the government. Can we do primary care research that'll help save the hospital from being overwhelmed, really? So for PANORAMIC, you know, anyone up to the age of 50 could join if they had a positive test and were sick for less than 5 days and had a comorbidity. But anyone over 50, regardless of comorbidity, could join. And again, we were comparing an oral antiviral with usual care. Hospitalization was the primary outcome. And we stopped recruiting when we had randomized 26,411 patients. And the reason why we carried on, we originally thought we might need about 5,000 patients per arm, is that the event rate was much lower now that the population had been vaccinated. And again, that speaks to the point that I made originally, that don't take evidence from other jurisdictions, other places with different sorts of people, and apply it to a very different setting. Because those pivotal drug trials from the companies were done in unvaccinated populations in settings that might not be completely comparable to where we were thinking of deploying the drug, the intended use population in the UK. So we recruited through general practices. We had 65 hubs where they took referrals from neighboring practices, and 4,500 eventually contributed. But then we also had that central recruitment where we had clinical staff phoning people up who had registered online. And we had a kind of algorithm that could search the primary care electronic records for people who had registered a positive test. And then we could text them to say that you might be interested in the trial. And this is just about longer term follow-up data, because, as I say, we're following people up over the longer term in PANORAMIC's case for 6 months. And we can suck data out of the routine record to get information, for example, about resource use and so forth, and hospital admission. And because we can confirm hospital admission, which was the primary outcome, from both the GP record and from the patient and from routinely collected data, we sort of managed to get ascertainment of over 97%. You know, we had lots of buy-in from, say, the Zoe app and pharmacies and social media and so on. And, you know, it took quite a combined effort, which included the National Institute of Health Research. They've got a clinical research network, mainly hospital-based, where they have funded nurses to help with trials and research. And we were able to get some help from them for primary care, well, considerable amount of help, really. Yeah, we focused on making this low impact for the patients, and they filled out their online diary, and we could phone them if they didn't, and they could have a trial buddy or trial helper that if they were too sick to fill in the survey, somebody else could give us the information for them. And that's really the recruitment. You know, we were really, I think, the fastest recruiting trial, to my knowledge, in primary care ever of a drug, while we were able to recruit into molnupiravir, which is a very, there are very few exclusions and very few contraindications. And then the small little bars there are our recruitment into the Paxlovid arm, which is a whole different kettle of fish. Many exclusions, many interactions because of the ritonavir in there. Nevertheless, we did manage to report just before Christmas then on the molnupiravir. And, our sample was well balanced between the two arms. One thing that we were quite proud of was the minority ethnic group representativeness, which is similar to national proportions for the population age group in the study. We didn't have enough people in the most deprived areas, so that's a bias. But it's a highly vaccinated population. We got the drug to people within 5 days, and we randomized them a median of 2 days after symptom onset, and over 90% completed the course, or so they said. Now, we found exactly the same proportion of people randomized who between this drug molnupiravir and usual care, who were admitted to hospital. So that you can't put a cigarette paper in between those two figures. So what we found here is, to answer the government's question, if you want to use this drug to keep people out of hospital in a vaccinated population, don't. However, the drug was safe. But what we did find was that it helped people get better quicker by quite a lot. In fact, the median time to recovery was 9 days with those who took the drug, and 15 days who didn't. And if you take into account a whole complex model, that amounts to 4.2 days, but the observed benefit is a 6-day difference. Furthermore, we found that people who got better stayed better more often, because it's a bumpy ride with COVID, isn't it, when you recover? And that bumpiness smoothed out if you took molnupiravir. And all kinds of secondary outcomes, including fewer contacts with general practitioners, favored the drug. And there you can see a really 4-nation study. That's where the people came from. So we did manage to sort of turn up that inverse research participation law. We looked at whether there was benefit distributed preferentially to certain subgroups, but actually looking at all the subgroups, there was no real group of people or people with certain characteristics who benefited more. But I just want, and similarly with recovery, and I just wanted to mention the virology results. So in a subgroup, we looked at virology, and we found, to put it simply, that people who took the drug had a lower viral load at 7 days, and fewer of them were secreting the virus at that point. But I will say this, that we found by day 14 that actually the viral load in the molnupiravir group was higher, and there was a little bit of, we're looking at this in great detail now, but on day 14, the significant difference between the groups disappears, and something's going on there about the virus, which we're looking at really closely now. In summary, this drug didn't reduce an already low admission rate for people who were vaccinated, but did recover. They did recover earlier. There was a bit fewer health seeking, and but the virology backed up the patient-reported outcome. But there was something going on there on day 14, which we'll tell you more about. So, yeah, lots of ongoing challenges to do this kind of research, which we can sort of speak about. But and other countries that are now taking the protocol and doing a similar trial there, for example, in Canada. But if I had to say what the biggest message here for me is, that research in primary care needs to be mandated, resourced, and embedded as part of a normal NHS care. And we need to be able to have that infrastructure ready for any epidemic and pandemic virus or variant of concern that emerges, and we can't take things for granted. So just go back to the blue plaque theme from St. Mary's to the Radcliffe Primary Care Building. So our nurses, Karen and Billy, put up this actually paper plaque in our trials unit, because they randomized the first patient into this trial then. And, we shouldn't take our foot off the gas. We got to be ready, I think, at all times as part of our pandemic preparedness to do trials like this. And just mention here that bird flu is being identified in mammals, as you probably know. So that's the sort of trial team. And thank you for listening. So we can maybe have a bit of a discussion.
Fantastic, Chris. Thanks very much indeed. I mean, just a beautiful talk, incredibly easy to understand what, it's, as you, obviously you're showing it and it all sounds relatively easy, but I know it was many sleepless nights. And as you say, a very, very intense COVID fueled for yourself period in trying to bring that forward. I know Gary wants to ask a question, but I'll, so why don't actually, Gary, I can wait. Why don't you go ahead?
How's it, Chris?
Hi, Gary. Nice to see you.
Long time no see. Amazing work, and it was just fantastic to see how quickly the UK responded, and how, I suppose athletic is the word the NHS proved to be, despite all the criticisms. I think few countries could really match that, as was shown by the fact that few countries actually did. Thousands of questions, but the one I wanted to pick up on is, when influencing guidelines, if you don't show a phase 3 randomized controlled trial, you struggle.
Yeah.
The placebo-controlled trial has become, ever since the scurvy days, the absolute reference standard for all gathering of evidence. And you said you would be happy to take a question on that, but that really is the question: is how do you influence policymakers? It's okay to say, okay, we've done this 30,000 trial in the UK, and in the UK, that doesn't seem to save hospitalization, therefore we don't need to do it. But if you want to change international guidelines everywhere, is it enough? Is it strong enough? And maybe linked to that is, have you looked at your trials compared with standard of care versus placebo? Is there a way that you can mathematically look at that and see how much placebo bias there is in an infectious disease? So a long question, sorry for that.
Yeah, no, I mean, it's so important, because I didn't go into it, but we showed that inhaled budesonide benefited people, in my view, in that principal trial. And there was a national expert committee in the NHS which looked at this evidence and said, we're going to use this stuff. And why wouldn't it work? I mean, Recovery had just said, in the sicker people, systemic steroids helped. And, in the less sick people earlier on, steroids going straight into the lungs. I mean, it had a mechanism. But suddenly NICE stepped in and they said, look, your primary outcome is patient reported, therefore it's subjective. And secondly, you don't have a placebo. Now, going to exactly the point that you make, so that they said, we need a placebo-controlled trial, and this drug wasn't deployed. Now, rightly or wrongly, I don't, but it goes to your point that if you want to change practice, as far as the regulators or the guideline people are concerned, you have to have a placebo. Now, I would say that is true, particularly in an early phase trial. You don't want to be taking stuff into pragmatic research that isn't got a good proof of principle. So the other thing is that obtaining placebo is very difficult. So they're kind of procedural reasons why you might want to kind of get kind of big scale evidence quickly in pandemic. so if you can't get a placebo, then, you know, do the open trial, and you can rule out drugs, as we did, for example, with colchicine and the antibiotics. There's 3 possible results with a trial like this. If you find no effect, the regulators are happy. You can see we find no effect with the azithromycin doxycycline. The regulators say, well, don't use it. There's evidence from the principal trial. So they believe you where there's no effect. They believed us with colchicine, which actually tended towards harm. But as soon as you're showing a benefit, they say, well, there's a placebo. The other issue that interests me here is the nocebo. A lot of people are also saying, yeah, I'm taking your sugar-coated pill, but it makes me feel terrible. So that somehow also needs to be taken into account in interpreting placebo-controlled trials. And I'll just make one last point about this, is that we are, so the EMA, European Medical Association, the FDA, that they won't accept a trial like this as part of their guidelines. But we are opening up another platform trial in continental Europe called ECRADE Prime, where we're looking at 3 arms, an active, a placebo, and then no treatment control. And we'll make an estimate there. So if we are able to carry on, get that trial off the go, we'll be able to put some numbers on that very question that you had. Sorry, I gabbled on too long there.
No, it's fantastic, Chris. Thanks. It's an interesting question. Thanks, Gary. So, Chris, you talked about the therapeutic alert that went out from Chris Whitty and his office. I mean, is there evidence from the UK that it actually did reduce prescriptions, and if so, how rapidly? And the second question around that is, you know, you also talked about context specificity. Which is, of course, key. But how should other countries, like South Africa, say, interpret your results in your setting? Is it good enough for us to then adopt in our setting? Because you're sort of, you know, you're almost making the point that it isn't.
Yeah.
What do you think?
Well, you know, I take the example of sore throat. Now, telling a GP in Oxford, look, you know, in general, don't give this drug to somebody with a sore throat. It's very different from saying to somebody in Day Hospital, Western Cape Metro, look, you know, don't use penicillin for this indication, really, because, you know, everything is different, really. It's the prevalence, the prevalence of complications. You know, I haven't seen a case of post-strep glomerulonephritis in 30 years, or however long I've been in this country, and hardly exists. All that does. So you can't translate sort of guidelines about antibiotic use, because the social determinants of health come into it. The bacteria are different, you know, the people are different, and the prior prevalence of complications is different. Now, how does that apply to, say, findings from this trial to your setting or these trials? The biggest difference that would come into my mind is the vaccination issue. You know, most of the people in this trial in the PANORAMIC trial had 3 vaccinations, and I'm not sure how that relates to the population that you might want to treat in South Africa. But you're probably more like the patients who were in the trials that the drug company did. And so that sort of study, the sort of pre-vaccinated population largely done in developing country, you might want to actually emphasize that sort of evidence and anticipate those sorts of effect sizes. But I certainly wouldn't say you've got to take panoramic findings and apply them to non-vaccinated people in South Africa.
Yeah, it's a difficult one, Chris, because as you know, we had a really enormous wave of infection as well. So there's the issue of natural immunity, even though our vaccination rates were in the 30%. our seroprevalence was in the high 95s to and higher. So it is difficult, and I appreciate that. I'll reserve my second question after the in-person group of somebody wants to ask a question. If there's anybody online who'd like to ask a question, please do raise your hand. Over to the seminar room.
Thanks. We've got a question from Graham. Hi, Chris. It's Graham Mankis here. Thanks very much for your talk. Just wanted to get to the point regarding the regulator and interaction between the trialists and the regulators. But one of the things in South Africa, we were very slow out of the blocks in terms of getting therapeutic trials for COVID started, and there are many reasons for that. On the side of the investigators, the regulators, other factors. But one of the important factors was the delays in getting approvals from regulators, and it was clear that in the UK that, based on your experience and the recovery experience, that there was incredibly rapid approval processes. And I'm just interested as to what kind of engagement had occurred with the regulators prior to the pandemic that allowed for those rapid processing of applications and approval that we could perhaps learn lessons from in South Africa.
Thanks, Graham, and nice to be in touch. I think the big difference probably was political will. So the government, through the chief medical officers, kind of issued, there's pandemic public health legislation that came into being, and that helped. But basically, when I was told that, I put in a grant application, same as everybody else, but when I was told that I'd been selected to lead this principal trial, I immediately got hooked in with the Health Research Agency. These are the people who organize ethics approvals. And I was able to have informal discussions. A particular ethics committee was allocated for this research, that they would scrutinize it, they would develop the expertise for ongoing scrutiny. And so I was able to discuss with the chair of that ethics committee before I submitted, tricky aspects around consent and so forth, ethical aspects. And furthermore, the MHRA, the Medicines and Healthcare Devices Agency, regulatory agency, I had people that I could talk to there. So prior to, within days, I was having discussions with these people. And I think that would have only happened because of government support and top-down engagement with the chief medical officers.
Yeah. No, thanks. Yeah, thanks, Chris. I don't see any other hands. So I'll ask just another question. I mean, your molnupiravir trial clearly, as you show, didn't show the benefit in keeping people out of hospitals, but did show benefit of getting to recovery quicker. So, in your discussions with the government and with the health agencies, I mean, what's your thought on who should use it in primary healthcare?
So, interesting one, because, you can, in a way, I sort of wish there was a really clear answer about this, but it goes back to that sort of 2x2 table. What's a meaningful benefit? And I think that is a contextual thing. So a lot of people are still getting COVID right now here in the UK, even people at similar, even people with a risk factor. Now, out of interest, the people with the highest risk factors with, badly damaged immune systems or transplants, stuff like that, they can get molnupiravir or Paxlovid directly from the NHS through things called COVID medicines delivery units. So a lot of people were saying, well, if those people can get it, everybody else or the next risk group down should get it. And in a way, we answered that question, actually, that, you shouldn't really expand the criteria for direct access. But look, this virus catches us with our pants down, if I can use that analogy, every time we look away. And should it happen that there's a new variant that bypasses vaccine protection and so on, and we're all in trouble again, and say the hospital is being overwhelmed, then you might have a case of saying, well, give this drug to key workers, for example, nurses, doctors, prison warders, policemen, transport workers, and so on, because we have shown it does reduce secretions, so it might reduce spread, but it also gets people back on their feet a whole lot quicker. So I think policy decisions will need to take into account context, but it costs £500 a course here. So that's not trivial. And my own view is that right now, you wouldn't want to be using this drug at scale, given the very low incidence of complications. And often showing when you shouldn't use a drug is often just as valuable, and in some cases more valuable than demonstrating, it works, use it at scale.
Yeah, there is another question, but you got to say long COVID, in terms of complications, but I'll leave that hanging for the moment. And again, there's the last question coming from the in-house group.
Thanks. Hi, Chris. It's Sean Basserman speaking. That was a wonderful talk. It's like breathtaking how clearly you describe these concepts and communicate them. So I've noticed that you've put quite a lot of emphasis on subgroups in trials. And people are, for reasons that I think we all know, wary about putting too much store on subgroups. You spoke about in your influenza trial, how you powered the trial to detect effects in subgroups. And I'm interested in how you do that. And then kind of related, I'm interested in how you think about heterogeneity generally, especially when you're using standard of care as a comparator.
Yeah. Well, I think, thanks, Sean. That's an important question, because I didn't really have time to go into the statistical aspects of this, but in a nutshell, we've used Bayesian statistics and not frequentist. So we have done numerous simulations, thousands and thousands of simulations of trials, where we've looked at the subgroups of interest that we think we want to power for, and then identified perhaps a number of possible drugs that we might want to put in the trial. And then what we do is to figure out what kind of sample size we might need to fill each of these cells. So say older people over 65, drug A, B, and C, and then you might have different categories. And so by doing thousands of simulations, you can see how each of those cells, how many people you need in each of those cells. Now, it's not like each of those is powered individually, because as the Bayesians will say, you can borrow information from neighboring cells to power up those individual comparisons. So the probability of benefit is increased given so you can use information from people who are a little bit similar to those in each of those cells for each of those drugs to increase your power. So long answer there, but it's through before you start your trial, doing numerous simulations around those subgroups, but using information from the trial overall to get that power. And then the issue of heterogeneity. Well, when you're getting, the efficacy trials where you have a kind of a pure population, if you like, with a single drug, and you really regulate that and you focus on them. And those are the trials where the GPs are saying, they aren't like mine, because mine are the great unwashed here. They're very different. So you got to do those trials before you start doing your pragmatic trials where actually heterogeneity is a virtue, where you want to see how this drug performs in the real world, because we've got so many examples from medical science whereby the efficacy trial shows a dramatic effect. Then you start deploying the drug in the real world, and suddenly, doesn't work so well. And that's because of the complexity and heterogeneity of the population. So doing the trial in the intended use population, warts and all, is the next step on, which is often forgotten about in applied clinical research. And so I see heterogeneity, in fact, in that type of study, the pragmatic study, as a necessity and a virtue.
Thank you.
Yeah.
Chris, it's just again been a fantastic talk. Really appreciate the time you've given us. Unfortunately, the lobby that you took Fleming's photo of Fleming's painting is the medical school and has just been sold. So if you want to see that painting, I'm not sure where they're going to put it, but it won't be in that building anymore, unfortunately. But it was again, as I said, a tremendous walk through a complex area, and just beautifully put. And it's we're honored, very honored to have you as a member of our division as an honorary professor, and really appreciate your time. And I think it is about time you also got over here so that we could all meet you face to face and have a braai and see you again. So thank you. Thanks, Chris. And
thanks so
thanks
much
to everybody.
Yeah, I was just going to say that. Sorry to interrupt, but yeah, great to be in touch with you again, and thank you for attending.
Brilliant. Thanks, everybody, and we'll finish now. Have a good weekend.
Yeah, thanks, Mark. Thanks so much,
Chris. Fantastic. You're a star, really.
The second,
it was beautiful.
Yeah, and I guess if